Okay, take 52 on a Sunday afternoon of This Is Our Video about competition between nitrogen fixers and non-nitrogen fixers. By our, I mean Elizabeth, Jacob, Chloe, and Frank, some of your ecology classmates. On the next slide here we have, on the left is an up-close shot of some of our pots. And we planted 2 seeds of each variety, that is borage, a non-nitrogen fixer, and crimson clover, a nitrogen fixer, into each plot. Still, there were quite a few of our trials where only one of the species germinated. This obviously got in the way of our experiment. On the right, you can see a picture of our entire trial, or our entire experiment. And we planted clover around the edges of our treatments in order to eliminate the edge effect, which is essentially a different amount of water or light based on being around the edge of the experiment. On the pink sticks, they are labeled both number of trial and how much nitrogen, that is pretty much our nitrogen gradient, how many miracle pellets we added to each treatment. Then the treatments of how many nitrogen pellets were all randomized in the experiment. Here's a better look at some of the numbers on the sticks. You can see those numbers are pretty random. We got a few 15s next to each other, a few 20s next to each other. That's just happening by chance. We use a random number generator. And also on the left you can see that the borage generally germinated before the clover. And on the right, the borage leaf is quite wide, which will eventually lead to problems we're going to see shortly. Here's another shot of the treatments. The front row you can see here, 5, 20, and control. Shows that neither plant was doing great in the 5. Both plants were doing awesome in the 20, and the control was kind of somewhere in between. Next slide we have, on the left, the lowest pot did not have a single of the 4 seeds we planted germinate. This was a problem because it essentially shrunk our sample size because we had no data for those pots that didn't germinate. On the right, the borage in the 2 pellet treatment is starting to really dwindle because of lack of nitrogen. Here's a picture of some of our pots on the day that we collected dry biomass samples. You can really see it's becoming an ecological system because we kept our pots and our different treatments too close together, and the borage was competing across treatments. Here's a final shot of our video. This is what it looked like last Tuesday when we harvested for dry biomass. Clearly, some of the more prolific borage plants were affecting multiple trials, pretty much altering how competition was happening in our experiment. Thanks for joining us.